Imagine a massive orchestra with 20,000 instruments, but only a few hundred play at any given moment, and the conductor constantly changes which ones are active. That's your body's cells. Each of your 37 trillion cells contains the exact same genetic blueprint, but they don't all act the same. A skin cell is different from a brain cell because different genes are turned on or off. But how does your body decide which genes play their tune and when? And what happens when the conductor makes a mistake? Everything is a system. Understanding how genes are regulated is crucial for grade 9 biology students. This lesson will explore the intricate molecular system that controls gene expression, from DNA to protein. Dr. Anya Sharma, a lead geneticist at the National Institutes of Health, explains that gene expression isn't a simple on-off switch. It's a finely tuned dimmer. At its core, specialized proteins, called transcription factors, bind to specific DNA sequences, acting like molecular switches. These factors, along with epigenetic modifications that change DNA accessibility, determine how much messenger RNA, or mRNA, is transcribed from a gene. But the story doesn't end there. The mRNA then needs to be translated into a functional protein, a process influenced by ribosome availability and the mRNA's own stability. Finally, the protein itself can send feedback signals, creating complex regulatory loops that maintain cellular balance. Our gene regulatory network system has 10 key components. We have 2 external inputs: transcription factor concentration, which is the amount of regulatory protein available, and epigenetic modification, which determines how accessible the gene is. The remaining 8 components are internal outputs: promoter binding affinity, mRNA transcription rate, mRNA degradation rate, ribosome availability, protein translation rate, protein degradation rate, feedback signal strength, and regulatory network stability. Let's look at how these components interact. A positive relationship exists between transcription factor concentration and mRNA transcription rate. More transcription factors mean more messenger RNA is made. Similarly, a positive relationship connects promoter binding affinity to mRNA transcription rate. The stronger the binding, the more efficiently mRNA is produced. However, a negative relationship exists between mRNA degradation rate and protein translation rate. If mRNA breaks down faster, less protein can be made. More ribosome availability has a positive relationship with protein translation rate, as more ribosomes mean more proteins are built. A negative relationship connects protein degradation rate to feedback signal strength, because if proteins are destroyed quickly, they can't send strong signals back. Lastly, repressive epigenetic modification has a negative relationship with mRNA transcription rate, often silencing gene activity entirely, like how some cancers evade immune detection. Build your gene regulatory network model and explore its dynamics. What if a mutation reduces the promoter binding affinity? Or what if a drug increases mRNA degradation rate? How does altering epigenetic modification affect the overall regulatory network stability? These complex systems are studied by professionals in exciting careers. Systems biologists and synthetic biologists design and analyze gene regulatory networks for applications in medicine and biomanufacturing, earning between $85,000 and $180,000 per year. Bioinformaticians computationally model these networks, helping us understand disease and develop new therapies, with salaries ranging from $80,000 to $160,000 annually. Everything is a system. Model it, build it, simulate it, understand it. Visit ModelIt KT12.com.